Dr. Dunn is an evolutionary biologist and writer, and he studies the diverse phenomena in our everyday world, from societies and networks of ants to the beating of the human heart. He writes about science in different articles and books, and pretty much anywhere that he can get a pen or a computer. He has written many books and regularly appears in magazines such as the National Geographic, Scientific American, New Scientist, and Smithsonian Magazine. He's written several books, and of note, those books are available at the University of Utah bookstore booth, where he'll also be signing books later on today. So we're going to hear his first of two talks right now, and so it's my pleasure to bring up Dr. Rob Dunn. Thank you.
Thank you very much. Everybody hear me okay? And do feel free to walk around in the back. I'd be walking around in the back. I can't sit still for very long. So it's an honor to be back here. I really enjoyed the last time I gave a talk here. I thought it was 2017. I didn't check 2013. Time has passed very quickly. I'm going to talk today about something that we call the human hologenome. And I'm going to talk sort of pulling threads from these two recent books, A Natural History of the Future, and then Delicious, The Evolution of Flavor and How It Made Us Human. And they may seem like they don't connect, but I'll connect them a little bit today. And for context, the hologenome, it's an old concept with sort of renewed interest, and it's the concept of the genes of the host. In this case today, it's the genes of the human and the genes of the microbes associated with that host together. And so it's a way of thinking about those collectives and also studying them in a holistic way. And today I'll be talking about the evolution of the human hologenome and the ways in which thinking about the evolution of these microbes and the human body together sheds new light on just interesting basic phenomena, but also applied consequences of those phenomena. And so I'm just going to walk through sort of various aspects of the human body and how they relate to this hologenome concept and how they've changed through time. And I'm going to start with stomach acidity. And if you read an intro medical textbook, an intro biology textbook, the dominant story about the acidity of the human stomach and stomachs more generally of animals is that it primarily relates to digestive functions. It's predigestion of proteins. But if you look across animals, one of the things that you see is that the acidity of the stomach actually varies very widely among mammals and vertebrate species. And so a number of years ago, we became interested, and this was led by Deanna Beasley, a postdoc in my lab at the time, in thinking about whether there was a secondary function of the human stomach, and in particular of the stomach's acidity. And what Deanna was really interested in was an old idea that had been floated in the literature in the 1930s and 40s, that an additional function of the stomach was actually as a kind of door to the intestines that influenced the ability of different kinds of organisms to get to the intestines. And so in this context, the extreme acidity of some kinds of stomachs would be viewed as primarily a way to keep foodborne pathogens from entering the intestines and causing havoc in the microbiomes of the intestines. And so what Deanna sought to do was then to compare different kinds of animals and see the extent to which the acidity of their stomachs could be predicted as a function of their risk of foodborne pathogens. And so what she predicted is that species that eat lots of food that is likely to have foodborne pathogens should have more acidic stomachs. And as you move towards species that eat foods that are far less likely to have foodborne pathogens, that you should see less acidic stomachs. Seems super basic, it was never studied. And what Deanna found was, in fact, this pattern. Try to remain conscious that on both sides here. And so when you look at obligate scavengers, typically very, very acidic stomachs, facultative scavengers, slightly less acidic stomachs. And then as you go all the way over to your right, you see foregut fermenters that are herbivores, and they have stomachs that are basically neutral. And so it looks like part of what's going on in the stomach, and Deanna's gone on to study this in other ways, is that there's this role of killing foodborne pathogens, and that there are adaptations that relate to specific diets. This becomes interesting as we zoom in a little bit and look at primates. And we've done this in part through literature searches, but also through a partnership with Sandra Anderson at the Center for Evolutionary Hologenomics in Copenhagen. And Sandra and a team there have found a pill that animals can swallow. It's an amazing new technology that records pH as the pill goes down. And so it allows us to record pH in the stomachs of many more mammals than we ever could before. And so we've done this now with some lemurs, with some baboons, and so we're starting to add more data from more kinds of primates. But as we do this, one of the things that we see is if we look at the omnivorous primates, in fact, they have slightly more acidic stomachs than do herbivores. And so in the case of the crab-eating macaque, pretty acidic. The herbivores are much closer to neutral. Chimpanzees, so far there's only been one study of one individual chimp stomach that was neutral. Not a great study, 50 years old now. We're very eager to get these chimps in the Copenhagen Zoo to swallow some of these pills so we get better data. But the remarkable thing you'll notice here is that humans have stomachs that are most like those of vultures, which is interesting in and of itself. It begs interesting questions about why this is the case, when did this happen evolutionarily. But the other thing it leads us to think about is what the consequences of this acidic stomach are in a modern medical context. And I would argue that there are several. First, because we know the pH of the human stomach becomes acidic very early in life. By age 2, it's sort of locked in. It should mean it's harder for adult humans to acquire new microbes from fermented foods, which appears to be the case. That many mammals have no difficulty in acquiring new microbes later in life. For humans, it's quite difficult. The other insight here is that any procedure or medicine that raises the pH of the stomach and makes it more neutral should increase the risk of pathogens surviving to reach the intestines. And we know this clinically, we know it empirically. And so these are insights that result from this sort of evolutionary perspective. And we can take this same perspective sort of down the GI tract to think about the intestines in general, and the large intestine in particular. And we can do so by studying humans in different contexts globally, but also by studying our living relatives, for example, chimpanzees. And so I've been part of a number of large collaborations to study chimp gut microbiomes across Africa. And so I partnered with a group at the Max Planck Institute in Leipzig in Germany, and they've sampled chimp feces from across this entire range. Made possible by putting up camera traps and observing where the chimps are. You don't have to habituate them. And so we then came in to take those fecal samples and to use genetic techniques to identify which microbes are present in them, to see the extent to which there's variation among chimp populations and what that variation tracks. And one of the quick things we saw, and I'm going to walk you through this figure a little bit. So on the x-axis, you have geographic distance. So as you go farther to the right, these are comparisons between chimps that are farther apart. And the y-axis, you have the dissimilarity of their gut microbiomes as a function of fecal studies. And what you see is the closer together chimps are in space, the less dissimilar they are in terms of their gut microbiomes. And this is actually a really strong effect. And chimps in the same community are even more similar. And chimps, if you study chimp babies, they're the most similar to their moms. And so there's this geographic effect on microbiomes, there's a parental effect on microbiomes, and there's some climate effect. And so this is interesting in a wild context. It's due to the mixture of vertical inheritance of microbes from mom during birth, environmental acquisition, and diet. And we can actually zoom in on the diet a little bit, which I'll do in a second, but it has the effect of engendering microbiomes that reflect local conditions. The other thing that we know happens is that one of the interesting features of chimpanzees is that they have gastronomic culture. And so chimpanzees in different populations use tools to do different things. And so, for example, Jane Goodall was able to observe at Gombe, her field site, that chimps would use sticks to collect these chromatogaster ants and another ant, the army ants. But what's interesting is that different chimp populations use tools to eat different things, and they have for decades. And so there is this persistent culinary culture, such that at this site very close to Gombe, where the same ants are present, the chimps there use sticks to collect totally different ants. They eat carpenter ants. And this has persisted now for 60 years. Well, one of the things this allowed us to do was to look at whether those culinary differences have effects on the microbiome by looking at these same sites. And what we found was, in fact, they did. Different cultures have different microbiomes superimposed on top of that geographic difference. Now, this may seem very obscure, but it becomes more interesting because this allows us to think about phenomena in humans, and for example, the fact that these connections between geography and diet can become decoupled when acquisition is interrupted. So, for example, C-sections or disconnects between ancestral and modern diets, and we see this in Western humans. And so there are the chimps again, and then here are data from the American Gut Project, of which I'm a small part, looking at humans. And so in humans, there's no longer any relationship in the United States between your geographic location and which microbes you have. And there's a much smaller relationship between diet and the microbes you have. And so we've disconnected these things. And so there's this disconnect that has many medical consequences. At the same time, we know that because of that disconnect, species are being lost that are not only locally adapted, but also key functional species. And as Justin and Erica Sonnenberg have shown, if you look at the Western gut microbiome, it's really a very unusual gut microbiome that's different from any other primate and different even from the gut microbiomes we would have had in the United States 200 years ago. And there are a whole suite of microbes that are missing that we didn't realize until very recently we were missing because of these disconnections. And so this broader perspective lends us a different lens in thinking about our microbiomes. We can do the same thing in thinking about skin. I've spent a lot of the last decade or so thinking about skin microbes. The skin, as you well know, is a habitat into and of itself. It has apocrine glands associated with hair follicles. These are sebaceous glands. It also has sweat glands, and then it has microbes that are in those glands, but also on the surface of the skin. And then there are bacteria and fungi and face mites. And sometimes when we study these organisms, we just see their history and how it reflects our own. So we've done a lot of work on face mites. We've shown that all adult humans we've ever sampled have face mites, typically 2 species that were until recently were thought to be each other's closest relatives. This guy here has them. There's no relationship between baldness and density. Thinking of myself, wasn't looking at you. But let's see if I can get this video going. Yeah, so these are on all your heads right now. We thought the 2 species on humans were each other's closest relatives. Studying them evolutionarily, we now know that they diverged 130 million years ago. And so they're interesting in and of themselves. Because they evolve more quickly than we do, we can use them to trace our evolutionary history. We can show that different human populations have different mites. And so this is just interesting. It's not informed our application too much, although there's one caveat I'll talk about later this afternoon. But then we can also study the bacteria and the fungi on the skin, which becomes more interesting medically. This is actually bacteria from the human belly button, amazing false color image from Martin Ogerle. And we spent a lot of time studying skin in general, belly buttons in particular. And the standard story, the medical story that most of you would have been taught, is that the standard skin microbiome is dominated by Staphylococcus. Some Micrococcus, also a little bit of Corynebacterium, but Staphylococcus is a big part of this story. And so we started studying this, but we did so in this evolutionary context, looking not just at humans, but also what this looks like in other species. And as we did, we focused in on these apocrine glands that you see on the far right, the twistiest blue here. They often get called sweat glands. They're really not sweat glands. Their function is to produce food for microbes that then metabolize those foods and produce the aromas associated with armpits. This is the sole function of these glands. In non-human species, this is clearly viewed as adaptive, and we know they play important roles in non-human species. We know that in gorillas can identify each other based on the smells from these glands. We know that armpit hair in humans and gorillas and chimps is a different kind of hair from the rest of hair, thought primarily to wick these smells. But we really don't know how different the microbes are in these glands in humans and other species, or we didn't anyway. And we also don't really understand their function. And now there's some work in primates about why these evolved in the first place. They may be a costly, sexy signal. And so you have to invest good resources to feed these microbes. And if you have a really strong smell, it suggests that you're healthy. It might prevent pathogens. There's some evidence of that. It could signal ill health. Fun to develop theory here. It's not really been developed yet, just a little bit. But we wanted to understand what the differences are. And so we partnered with Julie Horvath-Roth to study gorillas and chimps and baboons and rhesus macaques and humans, focusing on the armpits. And so the first thing we saw is that human armpits, for reasons that are still not clear, are much less diverse than these other species. But the other thing we saw is that there are major differences in which microbes are present. And so I want you to first focus on Staphylococcus in the yellow here. And so this is Staphylococcus and related species in the yellow. And what you'll see is Staphylococcus is much more common in humans than these other primates. In and of itself, that's interesting. And then the other thing to notice is that the relative abundance of Corynebacterium in gorillas and chimps is greater than in humans. Now, Corynebacterium is this sort of slow-growing, old-growth forest of the armpit. It accounts for most of the aroma that comes up. I mean, if everybody raises their arm right now, please. Right, so what's coming from your neighbor, a lot of that's from the Corynebacterium. Later in the day, it'll be a more biologically relevant example. And so what's going on here? Why are humans different? We started to zoom in on, well, what if part of this is product use? And so we did a number of studies comparing people who use antiperspirant, use deodorant, and don't use antiperspirant or deodorant. And then we also did studies where we subjected people to experimental use of deodorant or antiperspirant. And we could show that people who use antiperspirant or deodorant actually favor Staphylococcus bacteria. It's a very dramatic effect. And if you take people who are using antiperspirant or deodorant and you cease the use of antiperspirant or deodorant for a while, the Corynebacteria come back in. And so part of what's accounting for our medically normal skin microbiome is our product use, which is interesting in of itself. The other thing that's going on is that there's a gene called ABCC11, which controls whether you have dry earwax or wet earwax. The single nucleotide substitution, one genetic letter, has a huge effect on earwax. But that same gene also affects the apocrine glands. And so if you have the dry earwax version, your apocrine glands are tiny and more or less closed. And so people with that version basically don't produce armpit smell for the most part. They don't feed those microbes. And so what Kate did was to study the skin microbiomes of people with the two different versions of the gene, and she was able to show that that accounted for 40% of the difference person to person in how much Corynebacterium they had. And so these two things, product use and a single gene, have huge effects on the skin microbiome. And so what's the importance of this? Well, we know that people with different skin microbiomes are differently susceptible to particular pathogens. It's true for the coronaviruses. We know that skin microbiome composition influences attractiveness to mosquitoes. The more Staphylococcus you have, the more attractive you are to mosquitoes. So antiperspirants are actually making you more attractive to mosquitoes, which seems medically relevant. And we also know that whatever the ancestral function of skin microbiomes, we've radically altered them without thinking about it very much, which I think also becomes interesting. We can also think about vaginal communities, super complex microbiomes and huge human vaginal communities. And I was at a meeting a number of years ago when I was talking about microbiomes in general, and I met up with Elizabeth Miller and Beth Archie, who were studying vaginal microbiomes in baboons. And one of their observations, biologists do weird things, was that while human vaginal microbiomes are dominated by lactobacillus and are acidic in healthy women, that in baboons, there are basically no lactobacillus, and the vaginal microbiome is neutral. And so this raised the question that they hadn't really thought about because they were focused on baboons and not on humans, of what's going on here, why this big difference. And so we pulled together an evolutionary analysis of different species' vaginal microbiomes, and we're able to show that in nearly every mammal that's been studied to date, vaginal microbiomes are neutral. And that the exceptions are chimps, which are slightly more acidic, and healthy Western humans who have much more acidic vaginal microbiomes. And this is really due to the dominance of lactobacillus in humans compared to other mammals. And so something really interesting is going on here. I think it's medically quite relevant. We don't understand it yet. Part of what may be happening is that especially for Western populations on sort of starchy agricultural diets, that you're seeing more production of glycogen for these vaginal communities. And in fact, we see this if you look at the second to last column. Genital fluid glycogen to protein ratio is much higher in humans. If this is right, one of the things we would predict is that non-agricultural humans should show something very different. We don't know yet. And so something interesting is happening here that we don't fully understand, but obviously medically relates to lots of important phenomena. The fifth case I'll talk about here is that of what I call the extended hologenome. And so it's when we take our microbiomes and we co-opt them to do things outside of our body, and in particular, food fermentation. And so I do a lot of work on food fermentation, thinking about its global importance. And one of the things we know about food fermentation is it sort of falls within a general framework that was initially established to study brain size. And so when you study brain size in mammals or birds, one of the things we know is that one of the values of the big brain is that big brains allow animals to sort of buffer hard times. By having a big brain, you can feed on different species at different times. So think about a crow. You can store food. And so what we've also seen is that in climates that are very variable, you see animals evolve with big brains to be able to deal with that variability. As we contemplate a variable future, this is interesting in terms of our own brains and behaviors. But the other thing we know, and so the intelligence of crows emerges in this context, emerges where crows evolve in very variable environments. But it's also true for primate ancestors. But the other thing we know is that the other animals that do good in variable climates are animals with really variable microbiomes that can eat different kinds of things in different times just by fermenting them in their guts. And so the microbiome is a kind of gut intelligence. And if we think about our evolutionary history, by beginning to ferment food, we take advantage of both of these things. Fermentation makes inedible food edible, so some roots and tubers. It allows food to be stored. It helps to transform what is edible in a landscape, sort of changes the edible landscape. It may help kill foodborne pathogens, and it buffers hard times. And so in a way, fermented food is the interface between having a flexible gut. There's a giant mascot out there that's distracting to me. What's the Weber State mascot? What animal is that? Wildcat. Yeah, there it is. Here it comes. So this is a good point to pause and say that in nature, humans are not the only species that ferment food. And so some carnivores also ferment and store food. Foxes store food. Wildcats, I don't know if they store food, especially fuzzy wildcats. You can imagine it getting in the fur. But so fermentation is not strictly a human invention. I'll talk more about that in a second. But so fermentation is a way of being intelligent in two ways at once. And so one of the things that people long thought is that fermentation was really late in the human story. In the classic telling, we start to ferment food after agriculture. And so we start to farm, we have an excess of grain, we start to make beer, we have an excess of grapes, we start to make wine. It's now very clear that that's wrong. And one of the pieces of evidence that this is wrong is that as people have studied non-human primates, they've begun to see evidence of controlled fermentation in those primates. And so, for example, Liz Mallett has studied these capuchin monkeys, I read about this in more length than delicious, and shown that these capuchin monkeys often go to a specific tree with these giant fruits that you see here that it can't eat. They're too hard. And they knock hundreds of these fruits to the ground, and then they leave for 3 weeks. And then they come back after these fruits have rotted, and they're kind of like a sort of jungle kombucha, and they eat them then. And they do this repeatedly. And it's now pretty clear that they're intentionally fermenting these fruits so they can eat them. And so it begs the question, if these capuchin monkeys could do this, when could our ancestors begin to ferment? And here I need to introduce a sort of reality of fermentation and rot, which is if we're thinking about fruits, and to a lesser extent if we're thinking about meat, there's sort of two basic pathways to what happens when a fruit falls in the ground, or some grain falls in the ground. And one of the things that can happen is it can mold. And so fungus can take over the fruit, penicillium, for example, lots of other things. And when that happens, it's often not very healthy. It's often a very good vector for foodborne illness. But the other thing that can happen is that rot can be taken over by lactic acid bacteria, which produce acid and make foods relatively safe, and by yeast that produce alcohol and also make the foods, in some context, relatively safe. And so if our ancestors began to ferment foods relatively early, we might expect to see some evidence in our genome for the ability to detect and respond to good fermentation versus bad fermentation. And the last time I came here, we didn't know anything about this. And fortunately, science and technology has improved. And we now know that about the time of our common ancestor with gorillas and chimps, about 10 million years ago, that one of the things that happens in our genome is we get a supersized version of alcohol dehydrogenase that speeds up 40-fold relative to the standard primate alcohol dehydrogenase. And this is what we would expect if our ancestors were beginning to eat fermented fruits that were fermented by yeast and were alcoholic at about this time period. In and of itself, interesting, something clearly happened at this point. This is what makes alcohol pleasing and not just totally, well, it's just part of our modern alcohol story. The other thing, and it happens about here, the other thing that's recently been discovered is that humans have, in their fat cells and also in their immune systems, a special receptor for lactic acid that only a few species have. And when that receptor intercepts lactic acid, typically in the blood, it reduces the activity of the immune system. It sort of calms it down, and it stops the metabolism of fat. And it's thought that what's going on here is that lactic acid is signaling safe fermented foods and that there's food available, and so the immune system should not respond to the microbes it's intercepting. And also, you don't need to metabolize fat because there's sufficient food. And so that's interesting. That happens about right here. And then recently, we've studied the evolution of sour taste, and we've been able to show that about this same time, there's a change in sour taste from being aversive, which it is in most animal species, to being conditionally attractive. And so we like sour taste, or at least we're able to like sour taste relatively easily. And so all of this suggests maybe we are moving to fermented foods pretty early. The other thing we've started to see is archaeological examples of food fermentation that are far earlier than we thought. And so, for example, it's now clear that the first beer predates agriculture, and archaeologists are now arguing that one of the reasons we started to farm grains was able to produce more beer. And the first beer we've documented is a beer like this at Leo at Stanford is documented. And beer is certainly not the first fermentation because it's relatively difficult compared to a fruit fermentation. The oldest mead production now dates to 40,000 years. And then we get cool other stories from archaeologists. And so, for example, there are now a couple of mastodon finds where the mastodons look to have been butchered, pushed into a pond, tethered to the bottom of the pond with their intestines to a giant rock and left in the pond. And so archaeologists think these mastodons were being fermented and stored in these ponds. And the advantage would be you've killed a mastodon, you have 8 people in your family, even if you really love eating, there's a lot left over. And so archaeologists, being quirky people, Dan Fisher, who did a lot of the early work on this, had a friend who had a horse that died. He butchered that horse with stone tools. He put the horse in a pond, and then every month he went back and he tasted it. And he found that even after 4 or 5 months, it was still edible, and it had a flavor like Stilton cheese and steak all in one, which suggests it was a lactic fermentation. And he's not studied it microbiologically. We actually want to repeat this and see what happens with the microbes. The other thing we know is that these fermentations, although they happened outside of the body, were not totally independent of the body. There was a role for the body here. And this is a role that's sort of embodied in the term somat, which is a Korean word that means hand flavor. And it's a word I learned from my student, Emily Meineke, and her family, Joe Kwan, and Joe's mom, who everybody affectionately calls Mama Kwan. And Mama Kwan makes kimchi and describes the hand flavor of the kimchi as being, you can have good hand flavor or bad hand flavor, and it's what the person who makes the kimchi gives to the kimchi. And what we began to wonder, is this actually biological? Is it not just sort of a, mom's flavor, but actually mom's flavor? And so we wanted to study it with kimchi. Kimchi is a really difficult fermentation. There's a lot going on, hundreds of species. And so instead we started to look to the literature for what evidence there is that body microbes are working their way into fermentation. And so we know that some yogurts have gut bacteria from humans. We also know that some yogurts have vaginal lactobacillus. There are actually projects in my other home in Copenhagen to engage people in making vaginal yogurts. There are some projects that happen in Denmark that I can't take back to North Carolina. This is one of them. We also know that in washed rind cheeses, that the thing that reddens the cheese at the end and imbues it with many of its flavors is a skin bacteria lineage called Brevibacterium. Bacillus subtilis is in natto, the Japanese fermentation. It's alkaline fermentation. Lots of traditional Mesoamerican foods are fermented with mouth microbes. And so clearly there is this relationship on some level, but we wanted to study it in some more detail. Is there a body flavor? It's actually it's like a 20-foot tall sculpture. And so we decided to do this by partnering with bakers. And so we had bakers from around the world come to the Paratos Center for Bread Flavor, and we had them all make sourdough starters using the same ingredients, and what varied were the microbes on their hands and the microbes in their bakeries. And one of the things we could see is when they made those breads and then we smelled them and ate them, that there was an effect of the microbes in the hands of the bakers and on the microbes in the starter that we could detect in the flavor. A subtle effect, but it was there. And so there was the baker hand flavor. And then the bakers all wanted to fight over who had the best hand flavor. There was a lot of... But the other thing we observed, there was this total surprise, and so we didn't study it in the right way. And I'll back this up by saying, on most people's hands that have ever been studied, which is mostly Western undergraduate students with regard to microbes, there are about 3 to 6% lactobacillus on the hand and almost no yeast. When we study the baker's hands, some of the bakers had up to 70% yeast on their hands as a proportion of the fungi that were present. One of the bakers had 80% lactobacillus, even after scrubbing his hands. And so what's clearly happened is that they're changing the bread, but the bread is also changing them. And so there is this interplay between the body and food. And the other thing that's really interesting, and I'll pulling toward the end here so we have time for questions, and I know lunch is after this, which is a bad time slot. So stick with me just a little bit longer. Maybe the wildcat will come back for energy. Is we know there's an important interplay with olfaction. And so if we think about taste, I can't talk about this much length now, but taste is basically innate and instinctual. Sweet is sweet, umami is umami, sour is sour. To some extent, they modulate through life, but how they taste to you and how you think about them is mostly genetic. Olfaction is totally different. Olfaction, neuroscientists think, is almost entirely learned, or perhaps in humans, entirely learned. There's some debate. We could come back to it later. And in humans, it's actually very different from other species, because in most mammal species, most olfaction happens outside the nose. It's sniffing. The weird shape of the human head leads us to experience olfaction in 2 ways. One through orthonasal olfaction, sniffing, and the other through retronasal olfaction, what you smell in the back of your mouth. And so flavors are really dominated by a little bit of taste and a lot of retronasal olfaction. And so we have this very different flavor world than most other species. And Gordon Shepherd is the one who really pushed this idea forward. And what happens with this flavor world is that as you learn new aromas and flavors, they're categorized in your brain. And it's kind of like a card catalog system. This audience, how many people who still remembers card catalogs? All right, good audience for this. And so they get classified, and the more cheeses you smell, for example, the more kinds of classification for cheese you have in your olfactory system. But the other thing that happens is that those classified aromas get ranked as good or bad as a function of whether they're associated with good or bad experiences. And this can happen very quickly. And one way it happens is called the Garcia effect. If you throw up after smelling a smell, you learn it to be a bad smell in one go. And so very rapid negative valence. But the other is that if you're with your family, if you smell something again and again, it acquires a positive valence. And memories are attached to these good and bad smells. And so you can imagine as our ancestors moved around the world that this was really important to how they made sense of landscapes. And one of the really interesting things about this is that this learning happens, it begins in utero. And so great studies have been done mostly in France, where mothers are given different foods, and then the babies are studied with regard to their preference for the aromas associated with their foods. The most famous of these studies, half of the mothers were given, when pregnant, were given anise candies and half were not. And then they were checked for whether the babies did scrunchy-faced displeasure or lick-lipping pleasure at birth, at like 8 hours or something, really early. And what they found is that the babies whose mothers had eaten anise made that lip-licking, nursing face in response to the smell of anise at birth. The babies who did not did scrunchy-faced displeasure, and this has now been shown very rigorously to be true for garlic, for fermented fish, for vegetables, for cheese. And so you can think about a world where our ancestors move around the world, and in one generation they're learning new olfactory smells, they're learning new retronasal aromas and flavors, and beginning to appreciate what their family does. And so you can think about this in the context of fermentation. Fermentation can be so dangerous, but you can very quickly learn what's dangerous or not dangerous by using this smell library. The other quick piece of this that I find very interesting is that I've talked mostly about the hologenome of humans, or almost humans and other primates. But this is a broader story. And the fact that this is a broader story allows us to actually learn from the rest of nature and use it. And so, for example, here's this great painting by Carl Bloch. And one of the things to notice in this painting is that there's a wasp flying to the beer. And for a long time, people who brewed beer, people who made wine, thought of the wasps flying to beer as a nuisance. Recently, however, it's been documented that this appears to be the natural habitat of Saccharomyces cerevisiae beer and bread yeast. That in nature, they ride around in wasps, the wasps take them to sources of sugar, and the wasp use sugar that's a little bit fermented as an indication of sugar they might miss. And so it's a mutualism between wasp and yeast that we have co-opted to produce beer and wine and many delicious things. We figured out that we can use this to then go to nature to find new yeasts for beer and wine and bread, and we found many of them, including a yeast that's now produced commercially that can make a sour beer in a month rather than a couple of years, relying on our understanding of the hologenomes of these other species. We've also been able to show that those yeasts we're finding on wasps can make new kinds of bread. And then everywhere we look in nature, there's these kinds of relationships we can take advantage of if we want. For example, there are carrion beetles that find dead things, they roll them into a ball, and once they've rolled them into a ball, they put their babies, their eggs, inside the ball of meat, and then they inoculate the ball of meat with a Yerraria yeast that begins to ferment the meat so it's safe for their babies to eat. We found that some of these yeasts can be used to cure meat and are in fact related to the yeast we're already using to cure meat. And so these relationships are interdigitated. Why should one care about these fermentation relationships? I think we don't totally know yet. It's earlier days in the study of fermentation in the human body. But I think there's a suggestion if we better understand this, we may be able to better manipulate some of the problems that are going wrong with our microbiomes in ways that take advantage of these histories. And so, for example, Justin Sonnenberg and Erika Sonnenberg, who did a lot of the early work showing how many microbes we're losing from our gut microbiomes and their consequences, have recently begun studies on mice and also on humans to see if eating fermented foods can restore some of those microbes. And what they've shown is that eating fermented foods can restore the diversity of gut microbes. It doesn't restore the lost ones, but by restoring the diversity, it can restore some of the functions. And they can show that people who eat fermented foods every day, and mice that are forced to eat fermented foods every day, picture a little mouse eating kimchi, that they also see some remission of some of the inflammatory disorders associated with modern Western gut microbiomes from fermented food. Early days in this work, glad to talk more about it, but there's a clear indication that by understanding this better, there may be some things that we can do clinically in a useful way. So zooming back out, I've just picked and chosen a few cases of the ways in which thinking about this hologenome, humans and our microbes in this broader context, change some of the things we think about medically, in terms of well-being, and also just in terms of our stories of who we are. Every bit of our body can be studied in this way. The brain, as we understand better brain-gut linkages, is going to have this same kind of story. It's too early to tell. But other parts of the body, the same thing. And so I think as we look to the future, as we think about these evolutionary contexts, as we think about these relationships, we're going to see totally new things that change how we do our work with humans and how we think about wellness. And so I will close there. Thank you very much for sticking with me pre-lunch. It really is a great pleasure to be among you again, so thank you. Any questions? The gentleman with the face mites.
Is it on? I just wanted to first say nice haircut.
Thank you.
My question is, what is the perceived role of antacids and antibiotics in community? Is it affect the flora while people are taking them, but the flora reverts after they leave the system, or does it have more permanent effects?
So it was
Antacids and antibiotics.
So antacids, the understanding is it's a quick effect. The effect is greater if you're aging, so pH of the stomach becomes more neutral. As you get over 60, it begins to get more and more neutral, and so you're more at risk from foodborne pathogens. And often in a Western context, gut microbiomes by that point are pretty messed up. And so the risk of antacids is greater, and then just other things medically that alter the pH, but it's a pulse. And so if patients can think about reducing risk of foodborne pathogens in that pulsed time, the risk is very low. Antibiotics, if you start with a totally healthy gut microbiome and you take a normal course of antibiotics, most of the time you see what looks like recovery. Sometimes, for reasons we don't understand fully, even one dose of antibiotic can shift the gut microbiome in a more persistent way. And then we know if with repeated antibiotic interventions, you can start to lose key gut microbes and move to this sort of dysbiotic microbiome space that's really hard to recover from. And again, this is especially true in older people. And the challenge right now is we really don't know a good way to get those microbes back. I mean, fecal transplants have been really effective. It's interesting, when I talked about fecal transplants here in 2013, it seemed ridiculous. And as you well know, now they've become a pretty commonplace intervention for extreme cases and even non-extreme cases. But fecal transplants, they can restore health, but they're often still missing these key microbes. And so I think the other, and then the broadest way to think about this is that societally there's kind of a ratchet, that the more and more we use antibiotics, the more and more of these species we're losing from the whole system. And then you get populations where there's just no place to restore from. And there's a lot of work right now on how one might do those restorations. Restoring to health, there are clearly some good interventions. Restoring all the functions remains very, very difficult. I don't know if that answers your question. Yeah, thank you.
Very interesting. When we get mold on cheese and yogurt, should we be eating that, or is that all right?
So if you get mold on cheese and yogurt, the strains of mold that are mostly domesticated in those cheeses and yogurts are producing far fewer antibiotics than would a wild strain. And so they've been selected before we understood the microbes, the individual molds that are there, to be those that are producing far fewer antibiotics and far fewer toxins. And so in general, a moldy cheese is totally safe because of that long cultural tradition of what's happened there. If you get a sort of cheesemaker bro who's making his first cheese ever and doing it with wild microbes from his basement, it's less clear what's happening there. And so, so a great French cheese, thank medieval monks, safe. A basement bro cheese, I'd beware. Thank you.
So on my patient plans, whenever I give antibiotics or do antacids, I always recommend getting some probiotics along with it. Is there any kind of best practice? I tell people I don't have any brand snobbery. I don't care whatever your pharmacy has, get that. It's in kind of infant form or chewable, whatever. Is there anything I should be doing more specifically for when I recommend that, giving better recommendations on what they should take along with their antibiotics or with antacids?
Yeah, this is a great and tricky question. So as probably many of you know, probiotics are very unregulated. And so the relationship between claims on probiotics, is anybody selling probiotics here? Am I going to lose a friend? The relationship is modest. And so some clinically have been shown to be quite effective and some are not. And they vary greatly in how likely they are to establish in the gut. And so I think until we have a better regulation or a better understanding of which are which, it doesn't hurt to prescribe those probiotics, but know that there's going to be some variability that's going to be hard to control as a function of what specific microbes are there. The other thing I would say that Justin Sonnenberg's work is showing, and I'm looking in the wrong way, I don't see where there you are. is that fermented foods can have a lot of those beneficial effects too. And so there's a recent clinical trial that's not published yet, but shows that people who eat fermented foods every day, they're really showing a reduced risk of the effects, negative effects of antibiotics. And for most of these fermented foods, we don't know about any negative consequences. And so I think it's an easy additional recommendation. And I would just keep an eye on Justin and Erica Sonnenberg's lab if people are interested, because this is going to change very quickly. The other thing that's happening is that people are now moving toward probiotics that are more pharmaceutical-like, that are actually have a better chance of establishing in the gut and are producing specific compounds. And so in a clinical setting, if I come back in 5 years, this will be a very common thing. And so that's the other thing to watch for. And so there are now bacteria in clinical trials that produce serotonin in the gut and are being used to treat PTSD. In mice, they clearly work. There are bacteria that produce some of these key compounds that sort of relax the immune system. So that's the other thing that's going to be changing pretty quickly. Whether the regulatory framework can think about how to think about those things is going to be interesting. But we're at this turning point where we understand enough to move into safer things. And I see there are more questions. Just quickly, the other thing that will come on market soon is gut microbes that people can take that produce more lactase and that do establish in the gut that are going to allow adults without lactase persistence to drink milk as adults. So that's coming. And so if that's sort of a measure of where we're going, changing very quickly. I'm sorry. Questions? Yeah. I didn't mean to go on so long.
No, it wasn't that. It's just, maybe I misunderstood, but at the start you're talking about the acidity in the stomach and how after about 2, it interferes with the ability to establish this. So how does that comport with this idea of probiotics?
So part of it's dose dependent. So if you eat a lot of them, the probability that some are establishing goes up. Part of them are pill-like probiotics, where you're actually doing timing to increase the probability it makes it through the stomach. And I think that's also part of why you're seeing fermented foods having some more beneficial effect, because you're getting this daily dose that just increases the odds that some are going to establish. Yep.
Is there any research or science behind the idea of why bacteria is tolerated in some parts of the body but not others? I mean, E. coli is in our gut, but you put that in the urinary tract, it causes disease. You know, staph lives on our skin, and if you put that in the central nervous system, it causes disease. Why microbiomes in one part of the body is perfectly tolerated and good, and the other part, it causes disease?
I mean, from an adaptive perspective, there are some parts of the body where the control of microbes becomes really important. A more sterile-like environment is far more likely to be beneficial, and so the immune system invests in a different kind of control. The other part of that question is what allows the immune system to be so conditional, to respond to a species in one place one way and another place another way. And there's a paper that just came out this year that shows that depending on where they are, like E. coli is a good example, that it's producing different compounds, and so part of the response isn't a function of what compounds are being produced. But I think this is like still a pretty open area. And the other thing that's changed is our thinking about how sterile those other environments are. I mean, we used to think they were totally sterile, and now the sort of background blood virome is now known to be super diverse. And so I think that landscape is changing. But I don't have a great answer about conditionally, how is the immune system negotiating this balance? But it's a good question. Yeah. Oh, I'm sorry.
Is there any difference in the microbiome within a community of people who grow their own food versus rely on getting their food at the grocery store?
Yeah, so that's a great question. So the hypothesis in the literature would be that people who are interacting with plant microbes more generally are shifting their microbiomes. And there's some work in Finland that shows this effect. And so in Finland, kids that spend more time outdoors and that have more kinds of plants in their backyard clearly have different microbes on their skin. But our sort of holistic picture of how that change looks is still pretty modest. And it's so dominated by studying college students that a lot of these sort of daily things that we would like to know how they affect us are getting missed. We do know that so for example, for skin microbiomes, that if you compare the average Western skin microbiome, there's a lot of variation, but if we compare that to hunter-gatherer skin microbiomes, all the Western skin microbiomes clump in a big clump. And so we vary, but we vary much less than we might have historically and that people do globally. Let's see, I'm having...
Just wondering, there's got to be differences in the amount of fermented foods that people eat in different parts of the world. Have we studied and compared those? And you may have just answered part of that.
People have studied it a little bit. This literature is still very Western biased. What would be one example? I mean, so we do see dietary differences in microbiomes that are pretty big. And so, for example, I'm not going to be able to make it all the way to the fermented foods because we just don't know yet. But in Japanese guts, Bacteroidetes plebeius, it's a common species of gut microbe. In Japanese guts, the strain of that bacteria has genes borrowed from marine microbes that allow it to break down algae and clearly are better enable those Japanese populations to metabolize algae being consumed in sushi and in other contexts. And so what we have right now are their sorts of case examples. Like here's a place where we see the difference. The big picture differences, we're still pretty blind to them. But thank you.
What are some of the higher value fermented foods for consumption?
So I can tell you what Justin Sonnenberg's study has found, which is that the two biggest effects are from those producing lactate and acetate. So lactic acid bacteria fermented things and acetic acid fermented things. They have different effects. And what they find is that it doesn't matter too much which kind of thing is producing the lactate or the acetate, but having the mix of them has beneficial effects. So think vinegar and yogurt as an example. But Justin's lab has done 2 of 8 studies globally on this. And so it will change as we look more broadly. And most studies are really biased toward Western fermentations. And so they're sauerkraut, they're yogurt, they're not fish paste, they're not a lot of the other meat-based fermentations. And so we're still just seeing part of them. But if you're balancing lactic fermentations and acetic fermentations, our current understanding is that looks pretty good.
Any more questions for Dr. Dunn at this time?
And I'll stick around. I know lunch awaits, and I don't want to keep you from that. But thank you very much.
Yeah.